Hi everyone, this video goes into details about amino acid structure and protein organisation to the level of detail that you need for the Advanced Higher Biology course. So this is from Unit 1 of the course, Cells and Protein, and it's just the last section of Key Area 2, and we're only covering a little bit of that section. We're going to be explaining how the amino acid sequence determines protein structure, describing the basic structure of amino acids and the importance of the R group, and then going into detail about the different levels of organisation in protein structure. First, we'll look at the amino acid, obviously very important in proteins, because as you should already know, proteins are polymers of monomers, and those monomers are the amino acids. Poly just means many, mono means one. So basically, a protein is many amino acids joined together. There are many different types of amino acids, I think 21. They all have a similar structure, but are slightly different in terms of the R group, which we'll go into later. Now, amino acids are joined together to make a polypeptide chain by peptide bonds. So poly, many, peptide bonds. The actual structure of the amino acid, they've all got the same basic structure, and they only really differ in this R group. Okay, so every other bit of the molecule is the same. Now, these groups can vary in size, in shape, in charge. It could be positively charged or negatively charged, which is going to give that charge to the amino acid. Hydrogen bonding capacity, so basically how many hydrogen bonds can it join onto, and chemical reactivity. Based on these R groups, we can actually categorize amino acids, and you need to know these categories. Okay, you can have the basic group, and they're positively charged, the acidic group, which are negatively charged, the polar group, which are actually a group that really like water, okay, and a hydrophobic group which really dislike water. Okay, the wide range of functions carried out by these proteins are a result of the R group. Okay, so the way a protein behaves, the molecules it can interact with, so the way a protein folds is based on the amino acids and the relationships between the R groups within the amino acid. The first group we're going to look at is the basic R group. These are positively charged. They contain an amino side chain, which is 1 nitrogen and 2 hydrogen atoms. You need to know the structure of these side chains, and you need to be able to look at that side chain and recognize that that's the amino side chain, so therefore it's a basic amino acid. Our basic amino acids, there's 3 of them, are arginine, histidine, and lysine. At Advanced Higher, you don't actually have to know the names of amino acids. However, you might get given an amino acid and looking at the structure, and you'll get asked, is this a basic amino acid? And you'd have to say yes or no based on the evidence that you see. Just a little reminder that basic is a reference to how acidic or basic something is. Now, an alkali is a basic molecule, so lower down in school we would have just called them alkalis generally, unless you did chemistry, but we actually mean a basic molecule. So that is a molecule with a pH above 7. So that brings us to our second group, which is our acidic R groups, and these R groups contain a carboxylic acid side chain, okay, and they're going to be negatively charged. So they've got 1 carbon double bonded to 1 oxygen, and then they've got a hydroxyl group as well. Now, there are only 2 acidic amino acids, aspartic and glutamic acid. So the next group we're going to look at is the polar group. Now, these are the ones that are hydrophilic. So they love water. They will move towards water. They're attracted to water molecules. And there are 6 polar R groups. We've got cysteine, serine, threonine, tyrosine, asparagine, and glutamine. The opposite of our polar groups are our hydrophobic groups. These are the ones that dislike water. They avoid water. They try to move away from water molecules. So they're the opposite of our hydrophilic, opposite of the polar group. There are 9 hydrophobic amino acids. There's alanine, glycine, isoleucine, leucine, methionine, tryptophan, phenylalanine, proline, and valine. And just to reiterate, you don't need to know the names of the amino acids. You just need to know the R groups. So, just to summarise what we've talked about when we're about the basic amino acid structure and the R groups, I'm going to go over them again. At this point is normally when the pupils in my class would maybe take some notes. So if you wanted to, you could pause through these slides and take notes as well. So, amino acids. The sequence of amino acids determines the protein structure. Proteins are polymers of amino acid monomers. Poly is many, mono is one. So one amino acid, if you join a few of these monomers together, you get a polymer. Okay. When you do join them together, they're linked by a peptide bond to form the polypeptide chain. Amino acids have the same structure, but they differ only in their R groups that are present. These R groups can vary in size, shape, hydrogen bonding capacity, and chemical reactivity. And this is going to determine how the protein functions. The classifications according to the R groups are basic, acidic, polar, and hydrophobic. So basically, if you look here, we've got either basic or acidic, so these are opposites to each other, but then we've also got polar, hydrophilic, and hydrophobic. Now these are opposite to each other. Okay, so if you try and remember that they come in pairs, then you can remember the opposites, then you'll remember all 4 of our groups. Remember, the wide range of functions carried out by proteins is a result from the diversity of R groups within that protein. So you could have a protein that is primarily made up of basic amino acids. Or you could have a protein that's primarily based on hydrophobic amino acids. Or you could have a protein that has an equal mixture of all 4 types of amino acids. Or you could have a protein where all the acidic amino acids are at one end and all the polar amino acids are at the other end. This is going to dictate the function of that protein and how it folds. Based on that, we're going to go on to protein structure now. Now, looking at this diagram, you will see there are 4 levels of organisation that we're going to be looking at. The first level of organisation is the primary structure, and that is based on the sequence in which the amino acids are actually put into the polypeptide chain. So the order of amino acids in a polypeptide chain determines the primary structure. Now we're going to look at a few secondary structures. So there's a few different shapes that proteins can take, and the secondary structure shapes are all based on hydrogen bonding along the backbone of the protein. So hydrogen bonds between amino acids will stabilise these shapes, and it all depends on the position of the specific amino acids that the hydrogen bonds are joining together. So the first one we're going to look at is the alpha helix. This is a spiral kind of staircase. 1 alpha helix, 2 alpha helices. Okay. The second shape we're going to look at is our beta pleated sheets, and they can be parallel or anti-parallel. So it's basically a structure that forms kind of these strips here that end up lying next to each other like a sheet. Okay. Now they can either be parallel or anti-parallel based on the terminal of the chains. We've got a C here and a C there. That means that they're running in the same direction, because they're either going to run from N to C, because we've got our N there, okay, N to C, or C to N. Now these ones are running the same direction because they both end in a C here, meaning that they're parallel. But if this one was ending in an N here, then they would be anti-parallel. Now anti-parallel is actually the more common one. And again, this structure is secured through the hydrogen bonding between the R groups of the amino acids. Now the last secondary structure we're going to look at is our turns. This changes the overall direction in which the protein's going in. Now, we don't actually know the purpose of turns, but what does happen is it allows interactions to occur with other secondary structures, so maybe a helix interacting with a beta sheet. Just to recap our secondary structures, we've got 3 of them: the alpha helix, the beta pleated sheets, which can be either parallel or anti-parallel, the anti-parallel ones being most common, and also turns. Now these shapes are all stabilised by hydrogen bonds that are occurring between the different R groups. We're now going to look at the third level of organisation proteins, the tertiary structure, and this is all about folding. When our polypeptide chain folds, it's due to interactions between different R groups. Now, we've only really spoken about hydrogen bonds as an interaction, but in actual fact, there's quite a few other ones. There's hydrophobic interactions. So remember, we've got some R groups that are polar and they love water, and we've got other R groups that are hydrophobic and really dislike water. So all those polar hydrophilic R groups, they are going to be pushed to the outside because they're being attracted to the water. And remember, all the cell structures and the cytoplasm, they're in a very water-like fluid. So there's going to be water inside the cell. So the proteins are going to push all the amino acids that like the water, they'll be pushed to the outside, and all the hydrophobic amino acids, they're going to end up being dragged into the middle of the protein to try and get away from the water. Another type of reaction is ionic bonds. So that occurs between R groups that are positively charged and then another R group that's negatively charged. So the opposites are attracting. So that will make certain folds in the protein chain too, as the R groups come together with their interaction. Now our third type of interactions are London dispersion forces, and that's between R groups, the electrons within the R groups, when they kind of move apart, because electrons are constantly moving around, when they move in certain positions, it might bring those R groups together slightly, but then as the electrons move again, it will push them apart. So these are temporary interactions. Temporary. They're not stable because the movement of the electron is constantly changing. We can't have a stable interaction there. And then of course we've got our hydrogen bonds, which will still be evident. They're evident in their secondary structure. More hydrogen bonds will form in the tertiary structure as the protein chain folds over and secondary structures can be linked together with those hydrogen bonds. Our last one is the disulfide bridges, because there are some R groups that contain sulfur. So the disulfide bridge is a covalent bond that's going to form between 2 R groups that are containing sulfur. These are the 5 interactions that you need to know about for our tertiary structure, and they dictate the way a protein folds and gives it that 3D shape. Now the last type of structure is a quaternary structure, and not every protein has this. Only a small group of proteins have this. Proteins that have 2 or more polypeptide units will have a quaternary structure. And this describes the spatial arrangement of our units. Now if we look at this diagram, we have got in our collagen here, we have got 3 polypeptide chains sort of weaved together. That's a quaternary structure. Okay, but then when we look at hemoglobin, there are 1, 2, 3, 4 subunits arranged in this way where they're all sort of next to each other, clumped like that. That is our quaternary structure. Now you cannot get a quaternary structure if you have a protein that is just made of 1 unit, so 1 polypeptide chain. Another aspect to the quaternary structure could be the presence of a prosthetic group. Now a prosthetic group is a non-protein unit that's tightly bound to a protein, and it's vital for its function. I'll just use hemoglobin as an example now, but later on in the e-learning package, if that's what you're looking at from Balerno High School, you're going to have another PowerPoint that is focused on haemoglobin. But right now, I'm just looking at haemoglobin in terms of our prosthetic group. So remember, haemoglobin is made up of 4 subunits, but each subunit has a heme group attached to it, and this heme group is vital for its function. You will remember, I'm sure, that haemoglobin is responsible for carrying oxygen around the body, and it's the iron that's inside the haemoglobin that's actually responsible for carrying this iron, and that iron is present in our heme groups. So without the heme groups, we can't carry the oxygen around the body. So it's dependent on our prosthetic element. Now we're just going to park haemoglobin there. We're going to come back to that later. I now want to discuss some interactions of the R groups. So, these interactions shouldn't be a shocker to you that they're influenced by temperature and pH. We've been talking about temperature and pH affecting protein structure since National 5, so we're going to go into a little bit more detail. Now, if we increase the temperature, this disrupts the interactions that hold the protein together, and then the protein unfolds and becomes denatured. That shouldn't be new to you. But for advanced higher, what we're doing is we're tying that into the hydrogen bonds that occur between the different R groups. Okay, so the reason our protein denatures is because these bonds are disrupted at higher temperatures. So all the interactions that are bringing these R groups together, well, they disappear at the high temperature, meaning the protein loses its shape and therefore cannot function. A change in the pH will also affect some R groups, and they'll affect them differently, because remember, some R groups are acidic and some R groups are basic. So if we change the pH in the surrounding area, it's going to affect acidic R groups different from basic R groups. But if you increase or decrease the pH too much away from the optimum, then the normal interactions that are occurring between the charge groups are lost. So any of these ionic interactions, the ones that are occurring between our positive and our negative R groups, they're going to be lost, and then of course this will result in a conformational change. The interactions will be no more, so the R groups will not be getting brought together, so then of course our protein becomes denatured. So in summary, again, you might want to pause this and take some notes, but I'm just going to go over what we've discussed in our second half of the PowerPoint. Primary structure is the sequence in which the amino acids are synthesized into the polypeptide. So primary structure is the order of the amino acids. Hydrogen bonding along the backbone of the protein results in our secondary structures, which are our alpha helices, our parallel and anti-parallel beta pleated sheets, and of course our turns. The tertiary structure is stabilized by interactions between R groups, which could be hydrophobic interactions, ionic bonds, London dispersion forces, hydrogen bonds, or disulfide bridges. Quaternary structure only exists in proteins with 2 or more connected polypeptide subunits. They can also contain our prosthetic groups, which is a non-protein unit tightly bound to the protein, which is necessary for its function. The ability for haemoglobin to bind to oxygen is dependent on this non-protein group, this non-protein heme group. That is its prosthetic group. Interactions of the R groups can be influenced by temperature and pH. Increasing temperature disrupts interactions between our R groups, so therefore it changes the protein shape, begins to unfold, so it's denatured. Charges on the acidic and basic groups are affected by pH, so as pH increases or decreases away from the optimum, the ionic interactions between these charge groups are lost, so then the conformation changes, and of course the protein becomes denatured. Hopefully that will have helped you understand a little bit more about the structure of the amino acids. how that sequence of amino acids determines the structure of the protein, looking at the R groups and the interactions between the R groups to determine the 4 different levels of protein structure. Now, the following video that will come later is going to look more at hemoglobin and look at what happens to the heme groups when we change temperature or pH.